Good morning, everyone. Welcome to the class of biology on the fascinating world of biology. Hi, I am Nashad. I will be your instructor for this course. Through this course, we will learn about meiosis and its stages and importance. Here we will improve our reading skill. Now we discuss about meiosis. Meiosis. During sexual reproduction, two gametes fuse to form a zygote. The zygote divides repeatedly by mitosis to give rise to a new individual. All the cells in the young individual contain the same number of chromosomes. Thus, if the zygote has 4 chromosomes, all the new cells will have 4 chromosomes each. What are gametes haploid? The zygote is formed by two gametes uniting. This means that each gamete must have only half the number of chromosomes as the zygote. This is actually the case since the gametes contain only half the number of chromosomes as the parent cells which produced them. They are referred to as haploid cells. All other cells in the body are diploid as they contain the full number of chromosomes. Diploid cells in the reproductive organs gonads divide to form gametes by a process called meiosis. Since meiosis produces cells with the haploid number of chromosomes, it is known as reduction division. What is meiosis? Meiosis is a form of nuclear division such that the daughter nuclei produced contain half the number of chromosomes as the parent nucleus. In meiosis, the number of chromosomes remains half in daughter cell. Look here at hi mu. You don't look too good. You are right, sis. I feel like I am half the cell one used to be. That's cells calling to hi mu. Don't to look good like this. For convenience, meiosis is considered to consist of two divisions, meiosis 1 and meiosis 2. Meiosis has two phases, meiosis 1, meiosis 2. Each division in turn consists of prophase, metaphase, anaphase, and telophase. Look at this diagram and try to understand the process of meiosis. Diploid parent cells contain two pair of chromosomes, one pair, another pair. Replicate of chromosomes, replicate meiosis 1 and divide into two chromosomes here and two chromosomes here. Each daughter cell contains two chromosomes, but each chromosome consists of two chromatids. Here is divided to complete chromosome divided into half here and half here. In meiosis 2, half here of air. Four haploid gametes, each containing two chromosomes. Daughter cells are formed and each has two chromosomes. That's a diagram to understand the process of meiosis. Meiosis 1 phase and meiosis 2 phase. The sequence below describes meiosis in an animal cell containing two pairs of chromosomes, 2n equal to 4. Interphase. During interphase, chromatin threads replicate, producing two identical sister chromatids attached at the centromere. This is not visible under the microscope. The pair of centrioles also divide. In the previous lecture, we will learn more in details the process of mitosis in which nucleus, chromosome, chromatid, centromere, centriole. That structure is called centrioles, and the small tubes around there it's called esters. This thread-like structure is called chromatin threads, and here the round shape is called nucleus, and the outer line boundary is called nuclear envelope, and yellow part of the cell is called cytoplasm, and the most outermost boundary is called cell surface membrane. An animal cell at interface. Animal cell at interface. Stages of meiosis 1. Look at these pictures and try to understand the whole process of meiosis. That's a cell of animals, and these thread-like structures are called chromosomes, and round shape is called nucleus, and chromosome, the center part, this dot called is centromere point, centrioles, and these microtubes are called ester. Outermost boundary is called nuclear envelope, and this white color part of the cell is called cytoplasm. Homologous chromosomes pair up synapses. Chromosome form female parents, chromosome form male parents. Female X chromosome and male Y chromosome. These are formed spindle fibers, and the cell spindle fiber forms attached at the point of centromere. Spindle fiber long thread-like structure is called spindle fibers. Nuclear envelope disintegrates. Nuclear envelope disintegrate. Formation of chiasma. Crossing over result in the combination of genes along chromosome. At the end of the chromatids, one chromosome, another chromosome cross and each other and mix the genes heredity into the mix. Chiasma point. Prophase. 1. During prophase, chromatin threads condense, coil, and shorten to become chromosomes. Homologous chromosome pair along their whole length. This is called synapses. Homologous chromosomes have this same shape, same genes, and same length. One homologous chromosome comes from the male parents and other from the female parents. Esters appear around the centrioles, which move apart to opposite pole of the cell. As the chromosome shorten further, each chromosome is now seen to consist of two chromatids attached at the centromere. Now look here at this table. Chromosome shape and length. Surf the internet to find out which chromosomes do not have the same shape and the length. As the chromosome shorten further, each chromosome is now seen to consist of two chromatids attached at the centromere. Chromatids of homologous chromosomes may cross and twist around each other. The point where they cross each other is called a chiasmata, plural chiasmata. The strength of their coiling may cause them to break and exchange parts, a process called crossing over. Crossing over produces new combinations of genes along the chromosomes. See gene cloning in chapter 19. The centrioles reach opposite poles of the cell. The nuclear envelope and nucleus disappear. Spindle fiber form. Look at the diagram and try to understand the process of prophase metaphase. Metaphase 1. Cell in large spindle fibers. Chromosome is inverted at the equator point. Now we discuss the phase of metaphase 1. Pairs of homologous chromosomes arrange themselves along the equator of the cell. The two chromosomes of each pair face opposite poles of the cell. Homologous chromosomes moving apart. These are homologous chromosomes moving apart means in opposite direction. Anaphase 1. Homologous chromosomes separate and move to opposite poles of the cell. Note that each chromosome here is still made up of two chromatids. Phorosis occurs and the homologous chromosome is divided into chromatids. Now we discuss the next stage of the meiosis 1, telophase. A nuclear membrane forms around the chromosomes at each pole. This is followed by division of the cytoplasm. The cytoplasm cleavage into two, producing two daughter cells, each with the haploid number of chromosomes. The centrioles replicate. Look at the picture, look at this diagram, and try to understand the whole process of meiosis 1, prophase, metaphase, anaphase, and telophase. Gametes are haploid reproductive cells produced by meiosis. They contain half the number of chromosomes as the parent diploid cell. Meiosis consists of meiosis 1 and 2. Meiosis 1 and 2 also consist of the prophase, metaphase, anaphase, and telophase stages. During prophase 1, homologous chromosomes pair up and crossing over occurs. Crossing over is the process during which chromatids of homologous chromosomes cross each other and exchange parts. The points where homologous chromosomes cross over are called chiasma. Those parts at the point where chromatids cross each other, that point is called chiasma. Anaphase 1 separate homologous chromosomes. Telophase 1 produced two daughter cells that have haploid number of chromosomes. Stages of meiosis 2. Schematic representations of meiosis 2 is shown below. Now we try to understand the process of meiosis 2 by this schematic diagram. Prophase, metaphase, anaphase, telophase. Prophase 2. The centrioles move to opposite poles. These are the centrioles moves to the opposite direction. Nuclear envelopes breaks down. The nuclear envelopes break down. Spindle fibers appear. These are spindle fibers. Metaphase 2. That point is the center point is called equator. Chromosomes arrange themselves along the equator of the spindle fiber. Equator of the spindle fiber. Anaphase. Anaphase 2. The centromeres divide sister chromatids, separate and are pulled towards opposite poles of the cell. That's process like a video moving into the opposite. One is here, one is there, one is here, then other is there. Telophase 2. Telophase 2. Spindle fibers disappear. Nuclear envelopes forms around the two daughter chromosomes at each pole. Cleavage of cytoplasms result in four daughter cells being produced, each with half the number of chromosomes as the parent cell. Phoro cleavage foro occur inside, and one is create and other is create, one is here created and other is here created. Four daughter cells are produced but have the half number of chromosome as the parent cell. Now we complete our the process of meiosis. Meiosis has two stages. Stage Prophase 1, prophase 2, prophase metaphase 2, anaphase 2, telophase 2. And other stage is prophase 2, metaphase 2, anaphase 2, telophase 2. And in meiosis, the number of chromosomes remains the half as the daughter cell. Look at this diagram and diagnostic and try to understand the whole process of meiosis 1. Look here and try to understand the process of meiosis. Develops spindle fiber long, shorten spindle fiber move to poles here. Interface and discuss here about the parts of the cells like nuclear envelope, cytoplasm, cell surface membrane, nucleus, centriole, esters, chromatid threads. Animal cell. Stages of meiosis 1 describe the structure of cell, thread-like structure called chromosomes, chromatids, centromere, nucleus, spindle fibers, and in this process, crossing over is also takes place. The point at which the crossing over takes place, one chromosome and other chromosome, the point where crossing over met to each other chromatid and exchange their nuclear material is called, the point is called chiasma. And then we discuss the about of prophase 1 and prophase, metaphase, anaphase, telophase. Then we go ahead to the study of stages of meiosis 2. We understand the process of prophase 2, metaphase 2, anaphase 2, telophase 2. Now we discuss about the importance of meiosis. Importance of meiosis. Meiosis produces haploid gametes. When a male gametes fuse with a female gametes, the diploid number of chromosomes is restored in the zygote. This maintains the normal diploid number of chromosomes in the species. Meiosis results in variations in the gametes produced. Variations occurred due to crossing over and also due to independent assortment of chromosomes. That's the importance of meiosis. Independent assortment of chromosome means one chromosome from each pair can combine with either chromosome of the other pair. This results in four different gametes being produced from two chromosomes. Since fertilization is random, any sperm fuse with an egg. Such variation in the gametes produce variations in the offspring. Look at the diagram and try to understand the process. Parent cell with two pairs of chromosomes diploid number is found. Capital A, capital B, small b. Here look, small a, capital B, capital A, small b, a, small b, capital A, capital B. Four possible types of gametes can be produced, each with two chromosomes haploid number. Meiosis produces different combination of chromosomes in the gametes. For each pair of homologous chromosomes, only one member, capital A or small a, enters a single gametes. Variations increase the chance of survival of the species during changes in the environment. When environmental conditions change, nature will select those organisms with favorable characteristics to survive, while the other die. Those that survive will pass on their favorable genes to their offspring. Thus, though individuals die, the species lives on. For example, you may have a slightly different set of immune genes from your classmate. This may make you more resistant to certain diseases. If we all had the same immune genes, then a single new infection would be able to wipe out the entire human population. How does mitosis differ from meiosis? Summarizes the differences between mitosis and meiosis. Now we are going to discuss differences between mitosis and meiosis. Here we will look at how differ mitosis from meiosis. Look here, this table is about mitosis and other one table is meiosis. Here we discuss about mitosis and meiosis in the differences. First one is mitosis. Daughter cells contain same number of chromosomes as the parent cell. Now we look at the first point of meiosis. Daughter cells contain half the number of chromosomes as the parent cell. First difference is that in mitosis process, the number of chromosomes is same in data cell as in parents. And in meiosis, the data cell have the data cell have half number of chromosome in its cell as compared to the parent. This is one difference between mitosis and meiosis. Pairing of homologous chromosomes does not occur. Now we looked at how that tell about meiosis. Homologous chromosome pair at prophase 1. The second difference is that in mitosis and meiosis is that in mitosis homologous chromosome does not occur, but in meiosis homologous chromosome processes occur. Point 3, no crossing over. In mitosis no crossing over. Then we look about that meiosis crossing over may occur. The third one is difference is in mitosis crossing over occurs in mitosis, but does a crossing in mitosis crossing over does not occur, but in meiosis crossing over occur. Crossing overs actually occurs, it is necessary because the exchange of genetic material is occur in chromosomes, so that the removal of harmful genes would be removed, struck off, and a suitable favorable genes occur in our next generation. The fourth one point is daughter cells are identical to the parent cell. Variations occur in the daughter cell. The fourth one difference is that in mitosis the daughter cells, the daughter individual are same like to the parents, but in meiosis the daughter cells is not same like parents. The fifth one is two daughter cells are produced from one parent cell. Two daughter cells are produced from one parent cell, and in meiosis four daughter cells are produced from one cell. That's a huge difference between mitosis and meiosis. In mitosis two daughter cells are produced from parents, but in meiosis four daughter cells are produced from the parents. Sixth one involves only one nuclear division. One nuclear division involves two nuclear divisions. And the last point occurs in normal body cells during growth or repair of body parts, and in meiosis occurs in the gonads during gamete formation. That's all the difference between mitosis and meiosis. Here we look about the importance and meiosis. Look at the picture and try to understand the process of meiosis. That's the importance of meiosis. The main purpose of meiosis, it's the exchange of genetic material is occur in meiosis. And look here about the stages of meiosis. To the stages occur here about in the like prophase 2, metaphase 2, anaphase 2, telophase 2, and then we go to the study of my stages of my meiosis. Meiosis 1, meiosis में prophase 1, metaphase 1, anaphase 1, telophase 1, And that's the schematic diagram. Try to understand the process of meiosis and look at the diagram to understand the process of whole meiosis. I hope this lecture is very useful for those learners which are very poor in their reading skills. No, we will meet tomorrow in our next topic to the study. Then take care. Allah Hafiz.